Hello everyone and welcome to our second video of this first lab. And in this video it's going to be a bit longer and we're going to go through the language of anatomy, why it's important, and then connect that to one of our first skills, which is microscopy. So let's get started. Now, the language of anatomy is the cornerstone of this course. If you do not learn the terminology or how the words are put together, it's gonna give you a hard time. But once you've kind of figured out how all these structures are worded and how they give you hints, it's gonna make your life just so much easier. Terms that you're going to use in this class are derived from either Greek or Latin. We do also have some Arabic influence as well. But these words are put together in such a way that the prefix, or the front part of the word, or the suffix, the last part of the word, connect to a root that will give you some sort of understanding as to what we're talking about. So an example would be histology, Histology. The prefix there, hist, refers to tissue, and then, or the root there, hist, refers to tissue, and the suffix ology just means the study of. So we're going to be going through histology, which is the study of tissues. You're going to find a lot of root words that are connected in different ways with either suffixes or prefixes or two root words put together that are going to create this basis of the language that we're going to use in this class. Now, I am not going to read all of the terms to you and tell you what they mean. In your structure list that you guys are going to be getting, or packet you're going to be getting in class this week, I have a table that has all of the body regions you're responsible for. Your directional terms, your body cavities, your abdominal regions and cavities as well. All of this information is also listed in your lab manual. It's part of your required reading that you'll be doing. And I also have a Quizlet for you, so you can see just how important this is. We're also going to go through it in lecture too. So really make sure that this is your focus for this very first week of class. Many focuses, but this is going to be important. For those of you that are watching these videos that have not been added to the class yet, don't worry. On my webpage, you do also have access to that anatomical terminology Quizlet, so you can at least sort of try to be on the same level of everyone else in case you have. Now, how do we connect the terms that we have to learn to looking at a microscope slide? Well, when we make these thin slides of tissue, we can cut them a bunch of different ways. This is something that I used to do in my undergrad. We would take an organ, and in this case, our organ is an apple, and we would embed it or surround it with a certain substance. Sometimes it was really hard, like aerolite. Other times it was soft, like paraffin wax. And what we would do is we would encase it in a cassette and then slice it super thin along a different type of plane, depending on what we needed to look at. Oh, the thing here is, the hardest part about histology is not looking under the microscope and seeing the thing. It's trying to figure out what direction of the apple did you cut to give me this view. You can cut an apple a bunch of different ways and it will look different each time. So if we did equivalent of a mid-sagittal section, where we cut the apple down the middle, it would look like this. But if we did a cross section, it would look like this. Those look so different underneath the microscope. And without having understanding of the organ itself, of like, this is coming from an apple. Ah, that makes sense of why it looks like this. This is coming from a kidney or a heart. It's really hard to wrap your brain around what the heck am I looking at. So understanding your planes and how they're cut really helps you out with histology. When you're looking at your slides, a lot of times they will tell you it's a longitudinal section, so it's cut lengthwise, or it's a cross section, so it's been cut horizontally. Now in lab this week and next week, if you're like, I have no idea how this was cut, what the heck am I looking at? That's what I'm for. Please call me over and I can definitely explain to you either with models or words what the heck you're looking at. Okay? Now, we're going to take these organs, we're going to cut them along different planes, and it's going to give us a view of the organ itself. Now we have to look at, we need to take the information we just talked about, the apple, and how we cut it, and apply that to microscopy. Depending on how we cut a slide is how we want to look at something. Looking at cells, orientation, anatomical differences. And microscopy is the ability for us to look at those changes that we can't see with the naked eye. So here we're looking at 3 separate tissues: epithelium, connective, and muscular tissue. This first one we're going to look at later in our next video. But our ability to see this is because of a microscope. It's going to zoom in essentially and let us be able to see a whole new world. Now, if we move on to the next slide, what we see here is a microscope. Now, this is a beautiful brand new microscope. Our microscopes are going to look a little bit different, but overall, everything's pretty much the same. First thing I want to go through are the big parts for when you're carrying. We have our arm here, which you're going to wrap your hand around, and then you're going to put your other hand underneath the base here. Okay, the arm is basically connecting the base to the ocular and objective lenses, and then the base houses like all the electrical components for the light, etc. Now, the light is going to come from underneath the slide. Sometimes it's called a substage light, other times just called your light source. And we turn that on, obviously, somewhere on the base. And usually there will be a light control knob. This microscope has it right here. It's called a light control knob. The technical term and the term that I need you to know, which is listed in your lab manual, is the rheostat. The rheostat. And the rheostat is basically your dial that turns the light up and down, the intensity of your light. This microscope has it on the side. Ours is going to be on the front right here. And you never want it above 5 if you can. Between 4 and 6 is fine, but don't crank it up to 10. We have other ways of adjusting the light. So the light's going to come up from the bottom, and it's going to go through this apparatus here that we call a condenser. And what the condenser does is it focuses the light so it lands perfectly on the slide. And within that condenser, we have this little flange right here called your iris diaphragm. This is how you guys control the fine tuning of your light. So once you've set your rheostat to like 5, if it's too bright, you can close the iris diaphragm and make it dimmer. And if it's too dark, you could open the iris diaphragm and add more light. Typically, we only really play with the iris diaphragm when we're on higher powers, like 40x. Now your microscope slide is going to sit here on the stage, and it's going to clip in using this stage clip. The stage is movable, so it lets us move around on the slide, and the stage clips are what let us ensure the slide doesn't move. Okay, and how we move the stage is right here with our stage controls. One of these knobs is going to move the stage forward and back, and the other knob is going to move it side to side. And then you kind of use that in tandem to move around the slide to what you're looking for. When you're looking in the microscope, you're going to put your eyes up here to your ocular lenses, just like you're looking in a pair of binoculars. And you can adjust these to the width. So if your eyes are a little bit further spaced, you can open them up. If they're a little closer spaced, you can push them together. And you want to have both eyes open. It's very difficult, I know, but you want to have both eyes open when you're looking through the microscope. The reason is, in each of these ocular lenses, one will be clear, there'll be nothing in it, and in the other one, depending on the class you're in our class, we have a pointer, so you can rotate the pointer to the objects and you can say, hey, Miss C, what the heck am I looking at? When you get to micro, some of them will have an actual measuring tool in there for measuring the size of the bacteria. These have a magnification of 10x. So if you just looked at your ocular lenses, whatever you would see would be 10 times as big as it normally would. Right here is our revolving nose piece, and this is going to allow us to change the magnification. We look and increase our magnification through these objective lenses, ranging anywhere from 4 times to 100 times. And we'll go through that a little bit later. But that's a pretty quick rundown of your basic microscope parts. I did forget one thing. When we are looking through the microscope, we have to focus. And there are 2 focus knobs. We have our coarse focus, which is the big knob, and the fine focus, which is the smaller knob. Coarse focus is going to make the stage and objective lenses come together faster, so you focus faster. And fine focus is going to make slight adjustments in that differential. So we typically use that on our highest powers because we don't want to smash it into the objective lens. Okay, speaking of magnification, when we are talking about how big things are underneath the microscope, like what we're looking at, we have a couple ways of discussing it. We can just say the objective lens magnification or total magnification. I don't mind either, but there are other classes where they're going to be more specific about what you need to report. So each objective lens is going to have its own set of magnification. So again, scanning power is 4x, low power 10 times magnification, high power 40 times magnification. And how you get total magnification, which is how magnified everything is we have to multiply this number by the ocular lenses. Because remember, the ocular lenses also have their own series of magnification. So when you're looking at our lowest power, which is scanning, the object that you're viewing is 40 times its normal size. When you're looking at low power, it's 100 times. Or the total magnification is 4. So if I ask you for total magnification, I want this big number. If I just ask for like, what is the objective lens magnification, you want something more like this. Not specific at all, either or is fine. Now, something interesting happens when we are magnifying. Your field of view is what you see underneath the microscope. So put your eyes on those oculars. That circle that you see is your field of view. And we're going to talk about that a lot as we are practicing. But when you are on like low resolution, so it's not very clear to see, you're on pretty, you aren't going to see, you'll see a wide view of what's on the microscope slide, but you aren't necessarily going to see the exact thing that you need to as crisp and clear as you want. So the thing that I'm getting at is as you zoom in, you get closer and closer to the slide. And what that does is it narrows your field of view. So if this was us on scanning power, this little square right here is what we wanted to zoom in on. But this is what we can see. So if we center and zoom in on just that little square, that's all that we can see now. I cannot see any of this. So understanding that as we increase magnification, our field of view gets smaller is so important, because if you do not center your object before you move up in magnification, you're going to lose what you're looking for. So a practical example of this is just like zooming in on a picture on your phone. Like when you're taking a picture on your phone, you zoom in on the thing that you're looking at, but you lose all of the extraneous imagery behind it. And that's kind of what we're talking about. So for this next series of slides, you're going to hear this twice. I'm going to go through it in this video so you guys kind of have an idea of what we're going to be working on, and then you can always watch it again later when you get home after lab. Same set of procedures in class with the microscopes in front of you, so you can not only hear it again, but actually have the scope and the slides in front of you to practice this skill. So the first thing we do after we've plugged in our microscope and turned it on and have that rheostat on, and make sure there's light coming on, is we want to put the slide onto the stage and clip it in. Okay, again, we're going to put the slide here, make sure it's clipped in. Our clips look a little bit different. I'll show you guys what they look like on the board when you get to class. But we want to make sure that it's clipped in, and we want to make sure that we're on the lowest. Okay, you never, ever, ever want to just slide your microscope out when you're done at 40x. You want to not use a construction. Okay. If you haven't turned your scope on, you can do it. Typically, I have my scope on, but sometimes it's good to check about it. Again, as we talked about, you're going to turn your scope on and ensure that your rheostat is set to about 5. If you have to go to 6, that's okay, but what we do not want to see is having it go all the way up to 10. That blows the light bulbs out really, really fast, okay? Check your condenser, call me over if you aren't getting any light. I can kind of play around with some stuff. There's a few things that can go wrong that won't allow light to go through. One of the first things is when you're looking through your scope, If your objective lenses aren't clicked in, you won't see anything. But if there's no light coming from the bottom, call me over and I will help you with that. When you look through your ocular lenses, if there's no light, there's a couple places we'll talk about now and later that can help you with that. If you have to rotate your nose piece so that the lowest power is over the slide, if you haven't clicked it all the way in, you'll see black, okay? If you have clicked it all the way in, but you're still seeing black, you want to adjust that iris diaphragm. Sometimes that gets completely shut as we are moving the microscope to and from the cabinet. So just play around with that. Now, again, we want to make sure that we're always starting on the lowest objective lens. You never want to just have a slide and start it at 10x or 40x. And if your microscope, when you grab it, is on 40x, let me know. We want to practice putting our microscopes back at the starting point. And this is really important. It's a good skill to have because you're going to need that skill when you get into microbiology. So once we have everything on, we have light, and we're on our lowest objective power, go ahead and look through your ocular lenses, and you want to get everything into focus, okay? For the tissues we're looking at, they're going to be like a pink-purple color. In lab, for our practice, we're going to be looking at the letter E. So you want to use your force focus knob on the side of the microscope to focus it so you can see the letter E. Okay, or if we're looking at tissues, that sort of pink purple color. This is what you should see if you have completed everything correctly so far. One thing we don't really get into, you will more in micro, is the optics behind what you're looking at in a microscope. It tends to flip things upside down. So if you see an upside down E, that's totally normal. But it should be crisp and clear. Again, if you're looking through your oculars and all you see is white, you're going to want to use your stage controls to move around and find that letter E. Now, once the letter E is centered and focused, now you can rotate to the yellow objective lens, which is our low power. Okay, and at this point, we aren't going to really need to do too much in terms of focusing. You're going to want to use your fine focus to just adjust very minorly how clear that image is. Our microscopes are parfocal, so there's not a huge discrepancy in focusing ability as we move up in magnification power. So this is an example of what you would see at low power. Again, make sure you have some portion, maybe that bar of the letter E centered, so that when we go up in magnification, we're not just zooming in on light. So again, center your E or your tissue, whatever it is, and then you're going to rotate your nose piece to 40x. This gives students a lot of anxiety. It looks like it's going to touch. It's not going to touch. If we did everything right, it's not going to touch. This is also an area where we can have some issues. If you go up to 40x and you are turning that fine focus dial over and over and over again and it's not getting clear, take a step back and take a look at the closeness of that objective lens. It should be sitting right on top of your slide. If there is a large gap between the 40x objective and your slide, go back to low power, refocus, and try again, okay? With high power, light is going to get dimmer because the opening or the aperture within this objective lens is smaller, so it lets less light in. Adjust your iris diaphragm to add more light through the slide. Once you're on 40x, you do not touch coarse focus. You can only touch fine focus because we are so close to the objective lens. You're going to make some minute changes again with fine focus so it's clear to your eyes. This is what you should see. Okay, so this is what, if we centered on the black of the letter E, this is what we would see if we did it perfectly. Okay, and we're going to go through this, as I mentioned, together as a class. Now, if this were our tissues and we've zoomed in, we've observed our tissue, maybe we've taken a picture, we've drawn it. The next thing that you want to do is rotate your nose piece all the way back to the scanning power, okay? And then take the slide out. That way it's easy for you to start the whole process again. And for the next person as well. So that is it for this part of the lecture. I have one more for you guys going into epithelium, and I will talk to you guys then.